Today, that's good to record this. All right, Reza, you want to introduce Paul, and then we're going to hear about the cerebellum and the tongue.
Yeah, thank you, David, and thank you, everybody, for joining us. So we, in my lab, study eye movements and the role of cerebellum in control of them, as well as arm movements. But when you train animals to perform tasks, one of the things that you notice is there are other movements, of course. And one of the beautiful things about the marmoset is that it's an animal that we've been surprised by, and in terms of how apparently dexterous its tongue is. I sort of think that, you know, the way we use our fingers is not too far potentially from the way the animal, in this case, the marmoset, uses its tongue to forage for things. So, you know, we would use our finger to perhaps poke and prod in things. And you can't help but notice that marmosets are not only very visual animals, but they explore the world through their tongue. So serendipitously, along the vermis of the cerebellum, midline structures that get control of muscles from both sides are along the vermis, and right by the region that you find control of eye movements, control of saccades, you find control of the tongue. So like any good explorer, when we noticed this while we were recording from the cerebellum for saccadic eye movements, we thought, why shouldn't we also look at control of this beautiful other movement, control of the tongue? So that's what we did. And Paul has been leading this effort. Now, as you will see, the problem is really manifest in the fact that the tongue is this really complicated structure and has multiple degrees of freedom. So how do you track it? And how do you study its motion? And we're just beginners in this. We're just starting. Paul is a graduate student in biomedical engineering, and he's been leading a team effort that has included a number of other students, and he's going to mention them. But I thought to show you the work, because it's very much in its preliminary state, but we've made a bit of progress. So I would love to just share this excitement with you and see what you think. I'll let Paul take over.
Thank you so much, Reza. I'm going to quickly share my screen with everybody. If I can get a quick confirmation that you can all see it, that would be lovely.
Yes.
Okay, so as Reza mentioned, I'm a third-year biomedical engineering PhD candidate in the lab. And I've had just such a fun time really learning how to do cerebellar physiology in the non-human primate model that we use in lab, which is pictured here, which is the marmoset, that, as Reza mentioned, has quite dexterous control of their tongue. And thank you all so much for the attention that you will give me during this talk. And this is definitely a star-studded lineup, so I'm quite anxious about this, but let's get the show started. So all the work that I'll be showing you here coming up is really just it's made possible through contributions by the team at the Shadmehr lab. All of our members are pictured here, and really this is just a talented group that's been a pleasure to work with. So I'll quickly get into just a brief outline of what I'm going to be addressing in this talk. I'm going to start with a little bit of background information on the cerebellum and the tongue. I'll then describe to you some interesting things revolving around our common marmoset non-human primate models. I'll then go into a little bit of our methods. We spent a great deal of time in our lab building a neurophysiology rig. Specifically, I'll go into how we measure and quantify various behaviors, specifically eye movements and tongue movements. And then I will describe to you how we target regions of the cerebellum for neurophysiological recording. Then I will share with you some preliminary observations, in some of the Purkinje cells that I've been recording from, and how their behavior corresponds to licking behavior. And I'll briefly describe a little bit about how the marmoset licks during these tasks. So of course, it's without question that control of the tongue, fine control of the tongue in particular, is very important across a wide range of behaviors in humans and other species as well. You can imagine the intricate movements that take place in the mouth during swallowing, as can be pictured here. Or even during speech, you can imagine how difficult it can be for a non-native speaker learning a new language to even pronounce the right things due to being able to control their mouth in the right way and their tongue in the right way, to be specific. You can imagine rolling your R's, say in Arabic or Spanish. Finally, think of all of the times that you've chewed in your life, and then compare that to how many, hopefully few times you ended up biting your tongue. And then compare that to exactly how much time you spend consciously thinking about what your tongue is doing in your mouth. It's pretty staggering that we don't consistently bite our tongues off every time we chew vigorously. And one can only, as a cerebellar physiologist, think of how it is that the cerebellum contributes to this sort of neural control. And that's really what I'm hoping to get at over the next few years of my PhD. So to begin, we do know that the tongue is spatially represented across the cerebellum quite widely. You know, we can look at human fMRI studies that show bilateral activation, primarily across lobules 6 and 7, during a variety of different movement tasks. Furthermore, at the level of the deep cerebellar nuclei, there has been reports of a widespread yet unequal representation of extrinsic tongue musculature control across all 4 of the deep cerebellar nuclei in the work of Aldous and Bowman, and some of this can be fairly consistent to that of Thatch as well, as you can see here in his illustrations of the somatotopy of the deep cerebellar nuclei. Furthermore, what we do know as well is that dysfunction of the cerebellum impairs tongue control. And really, we can look at lesion studies, for instance, where widespread types or widespread causes of lesions, rather, can cause dysarthria. And this can happen really across the entire cerebellum, thinking of tumors, infarctions, hemorrhages, vascular malformations, and abscesses as well. Furthermore, there is disease that can, of course, also cause dysarthria, and of course, in its later stages, tongue fasciculations and eventual atrophy, such as spinocerebellar ataxia type 36. So really, there is somewhat of a link between control of the tongue and the tongue generally, as well as the cerebellum. So let me get into a description of our marmoset, our non-human primate model that is the marmoset, okay? So these are a very interesting small New World monkey that originate in Brazil. They have no known lethal zoonotic diseases, so they're fairly convenient to work with for researchers. They've recently gained attention in the last couple of decades or so for neurophys studies because of their potential for transgenic manipulation. This is because of their they have a very short gestation period. They regularly give birth to twins or triplets. This means a high breeding efficiency, which grants a great deal of potential for germline transmission of genetically modified models. Lots of promise in that aspect. Furthermore, in terms of behavior, they share a lot of similarities to us. They're very social and live in family units. They are vocal and they rely on a very impressive wide range of species-specific sound production to communicate. And they're also very visual creatures. They make saccades and they use all kinds of eye movements to explore their environment. Their brain is also rather interesting in that despite having a very smooth cerebral cortex, they have a very well-evolved cerebellum, and it's organized in a way that's sort of classic in that they have transverse foliation and longitudinal compartmentalization of their cerebellum that we've come to know and love. They do have molecular compartmentalization that reflects fundamental organization of the mammalian cerebellum, and in fact, their cerebellum is very similar to that of the macaque. They rely heavily on fine control of their tongue in a wide range of their behaviors, and I'll address some of these behaviors to come right now. First is in vocalization. They make again, as I mentioned, a wide range of sounds to communicate. Among these are trilled twitter calls, which are low-pitched with a distinct vibrato sound, and they fluctuate in frequency, as well as fee calls, which are loud, high-pitched whistles that are sounded in a series of 1 to 5 notes that last about 2 seconds each. Very complex sounds. In fact, if you were to walk into a marmoset colony with your eyes closed, you might just think that you've walked into the Amazon rainforest. It's really quite remarkable to listen to. These types of sounds are used in a host of different things, such as locating lost group members, maintaining group cohesion, territorial defense, and attracting mates. In fact, also, they use their tongue in visual cues, particularly for mating, where females, like in the picture, or the GIF rather, on the right, will flick their tongues to solicit potential male breeding partners. Finally, in feeding, which is, of course, a highly demanding of evolutionary basically driven by evolution, right? So these animals, 70% of their diet is plant exudates. And to get at these, they basically have to climb up trees, gnaw holes in these trees with their teeth, and then use their tongue to extract the desired exudates. Complex movements are demanded in sort of things like these sorts of behaviors. So I have two sort of broad research questions moving forward. The first is, how exactly is the tongue spatially represented across the marmoset cerebellum? With a creature that relies so much on their tongue, I can perhaps assume that there must be some vast representation of the tongue across different regions of their cerebellum. To get at this, I will perform acute extracellular recording in awake, behaving marmosets from subpopulations of Purkinje cells that are located across various regions of the cerebellum. And then the second question is sort of whipping out our microscope, is how exactly do Purkinje cells contribute to control the marmoset tongue, and what aspects of tongue movements are encoded in their simple and complex spikes? To get at this, I hope to characterize Purkinje cell responses during a variety of different licking behaviors and essentially look for patterns. So to get into methods, our lab spent, as I mentioned, a great deal of time developing a neurophysiology rig, and I'll describe it a little bit in the next few slides. Marmosets during our tasks are positioned in a chair that looks something like this. They are seated in front of a TV screen, and they are making saccades to targets on a TV screen, and we are tracking their eye movements at 1,000 hertz. For successful saccades, marmosets are given reward, and they retrieve these rewards essentially at their own choosing. They make saccades and then retrieve reward. Furthermore, we have a data acquisition system and a micropositioner that's essentially driving our probes and recording neural signals. And additionally, we have a photodiode that measures screen delay and an overhead lick camera that I use to record licking behavior and eventually quantify this licking behavior. So here's a little bit about the task design, basically what the animals do when they're seated in this chair and performing our experiments. First, animals begin by fixating on a central target. We present them with a secondary target from fixation that they then have to saccade to. And when we detect the saccade, we jump that target to a third target on the screen. And you can see this sort of in the eye trajectories plot that I have on the right over there for you. When they successfully make that initial saccade followed by the secondary saccade, we deliver reward to them, and they retrieve this reward through licks. We have two food tubes in our setup, which obviously forces the animals to make really a wide range of lick types to get at this food, depending on where the food is in the tube and how far it is from their mouth. So how do we measure tongue movements, and how do we essentially extract kinematics and quantify them? Well, we use deep learning made possible through markerless pose estimation with Deep LabCut. Deep LabCut is a wonderful open source Python-based package that allows you to essentially label frames in a video using a GUI and then train a deep convolutional neural network to track the tongue and other regions that really a user will mark across videos. It's really well generalizable, and it's a very powerful tool. Here's one example of a lick being tracked. As you can see, we have 4 points on the tongue that are being tracked. We have the tip, the midpoint, and then the left and right edges, which are at some middle point between the midpoint of the tongue and the tip of the tongue. We track the nostrils as well. I hope you can see those markers that are in purple, maybe a little bit hard to see from your end, but this sort of gives us a little bit of an idea of what the face is doing during these licks. And it's also a really good reference point since these animals are head fixated. In addition to this, we track the food tubes and the food in the tube at its most dense location. That way we know where the food is at, and we know exactly when the tongue is making contact with the food and such. With this, we are able to recreate the trajectories during these licks, as you can see here in this trajectory plot. And we can extract kinematics like displacement of the tongue, velocity of the tongue during licks, and the angle that is taken by the tongue during a particular lick like this. Furthermore, we can fit polygons to these markers and get an idea of what the surfaces are doing from this two-dimensional perspective. This is really useful, again, in tracking if the tongue has made it to the food, if the tongue missed the tube, if the tongue hit the tube but not the food. We can remain informed on what is happening in the frame of the video. So now how do we get to the cerebellum? We have animals that are doing a task, making saccades and licking, and now we want to record from the cerebellum. So this is how we go about it. We use imaging to guide the design of our alignment apparatus that essentially get us to regions that we're interested in the cerebellum, okay? So we begin with a subject-specific pre-op CT and MRI, as well as a post-operative. When I say post-op, I mean after we have implanted the head fixation apparatus into the animal. This post-op CT imaging includes a reference ruler that is eventually used to bridge our imaging coordinate system with our model-based computer-aided design coordinate system. We co-register these images to get a full representation of the animal's head, including its skull, the head fixation post, the brain, the cerebellum, and then we make markings using a software called 3D Slicer. It's a very powerful visualization imaging-based software. It's been really great for our lab. We take these markers and we import them into a model environment using the reference ruler markers to align everything such that we have a full representation now of the animal's head, skull, brain, and the markers that we've chosen in our model space. And with this, we design trajectory plans, as you can see here. So these trajectory plans then inform the design of mechanical alignment apparatus that we 3D print. This is one example of one of our pieces of equipment. This is called a grid guide. And essentially what we do is we are able to install this grid into the animal's chamber, and each hole corresponds to one region across the cerebellum. In one of our models, which is the one that you see here, we're able to target lobules 5 through 7 and about a 5 millimeter lateral range within these lobules. So we can remain informed and repeatedly target the same regions across sessions. We essentially align our entire stereotactic micromanipulator and frame to one of these holes, and we drive through into the brain with the probe that we choose to record with. We've recorded in our lab with 4 types of probes just so far: heptodes, tetrodes, and silicon array high density probes. So now when we're recording, how do we identify Purkinje cells? How do we know that we're recording from a Purkinje cell and not, say, mossy fiber input from granule cells or so? How do we know what we are targeting and if we should essentially record that cell? How do we know that we're not fooling ourselves with the neural recordings that we're acquiring? So, of course, the result of all of this is signals like this. I want you to pay attention to channel 3, which is up here in orange, or yellow rather maybe, and channel 5, which is here in orange, as well as channel 6. And what you can see is multiple units. So in this video, you will notice a fairly standard Purkinje cell signal. We have the high frequency simple spikes that are firing, the lower frequency complex spikes. And really there are 2 challenges when recording signals and trying to identify if these are in fact Purkinje cells. The first is, how do we know that any given signal on any given contact is from a single unit and not multi-unit activity? We do this by essentially relying on refractory periods and refractory period violations, which looks something like this. So if we were recording from a single unit, we would expect that the simple spikes have a refractory period that we then, that you can of course see here illustrated by this autocorrellogram. We can quantify this, and we do using this, this expression, this conditional probability essentially, which is the probability that at a given time 0 where a simple spike has occurred, that a simple spike will occur again at some time in the future. And what we expect to see is a refractory period, of course. So 5 milliseconds or so of no simple spikes firing, or a very low probability of simple spikes firing after that. That's how we identify that the simple spikes we're seeing are from an individual unit and not multi-unit. And also the second challenge is then trying to understand if the complex spikes that we're seeing in a signal and the simple spikes come from the same cell. This can be illustrated with this cross-correllogram, where we see a suppression of simple spikes that occur after a complex spikes. This is an intrinsic feature of Purkinje cells, and we rely on seeing this when we're recording. And we can again quantify this with the cross probability expression that you see here, which is that at time 0, given a complex spike has occurred, what is the probability that a simple spike will occur at some time t? And what we expect to see is roughly 10 milliseconds or so of simple spike suppression. When both of these conditions are met and not violated, we can then say, okay, we're recording a signal that is first single unit activity and from the same cell, being the 2 types of action potentials that we see. Then comes the challenge of spike sorting, which is really a challenge across the field. And it's mainly a challenge because of the difficulty and the diversity of trying to find complex spikes. Complex spikes can look can have a variety of different looks to them several characteristics that can include, long or shorter types of complex spikes, many or few spikelets. And so what we've done as a lab is we've developed a custom-built open source spike sorting software that is in its beta version right now that we intend on publishing about and distributing to the community as is demanded. And really its goal is to address this complexity of sorting complex spikes. And so we've put in a lot of time and effort into developing Purkinje sort, which is what it's called. And it's been developed by our lab mates, Ehsan, Jay, Amin, and Kyle, who are very talented software engineers. It looks something like this. It's a GUI-based supervised sorting software. It uses a host of different dimensionality reduction techniques and clustering techniques to basically make the process of spike sorting faster. And it's been proven to be really, really powerful for us. And what we've learned in sorting through just tons of cells is really that the quality of your analysis really depends on the quality of your sort. And so we make it a point to quantify the quality of a sort. We monitor these the suppression of simple spikes, for instance, and from complex spikes, as well as refractory period. We try and make sure these things are never violated and that we're extracting every complex spike possible as well as every simple spike possible. And we oftentimes as a lab will sort each other's cells as well and discuss techniques that we've used and the outcomes of those techniques to make sure that we're not losing out on any potential action potentials and having the highest quality sort possible. Now, here are some observations of some of the cells that I've recorded from in the cerebellum. Thus far, I've recorded, so first of all, let's just talk about task dynamics. What is the animal doing when it's doing the task, okay? So this marmoset behavior during our tasks. Well, the subjects will typically alternate between completing trials and then harvesting rewards. So they bite, and this is really up to them to choose. We don't put any constraints on when they can make saccades and when they can lick for the food tube. And what we see happening is basically that what I have plotted for you here on the x-axis is time, on the left, on the y-axis is the reward tube capacity that I showed you in some of the video frames prior. 0 corresponds to an empty reward tube. It's really just where the food is in a position inside the reward tube that the animal can no longer reach. I'm calling that 0 capacity. 1 is a full food tube. Above 1 is the food tube overflowing. And then in between 0 and 1 is, of course, the food somewhere within these bounds. This blue trace corresponds to the available reward in the food tube. Black stars are successful trials consisting of 2 saccades typically, and blue dots are trains of licks. And what you can see here is these dynamics sort of unfold in that the animal will complete some number of trials. It'll then decide that the food in the reward tube is worthy of initiating licks and stopping the trials, and they'll begin licking to retrieve rewards. And they typically lick in these sort of rhythmic bouts of licks that I'll describe a little bit more later. And what we see here is that the animals do indeed, when they're licking, stop making task-related saccades for the most part. What I have here is data from one subject. This left, I plot to you on the x-axis time from the lick that initiates a licking bout. On the y-axis, I plot for you the probability of a saccade onset of a task-related saccade. And as you can see here is that before licking bout is initiated, the animal's making task-related saccades and then stops when it starts licking. On the right, I have the same exact thing except aligned to the last lick of the bout. And as you can see, looking backwards 1 second in time, the animal has not been making saccades. And then when they complete the task, they will continue making saccades after that. And here I have just bar plots that plot to you the difference in the 1 second before initiation of a bout and 1 second after. And the same thing goes for 1 second before the end of a bout and 1 second after. And you can see probability drops, then probability increases. This is consistent for the most part across subjects. We have a second subject where we see pretty much the same thing. So there's an interesting decision that is made here by the animal that I'm going to exert effort to perform saccades. I will then stop, and when I think reward is worthy of retrieval, I will initiate a ton of licks, get that reward, and then continue making these trials. And this really just captures the dynamic, sort of unconstrained style of this task. So the licks that are produced are very diverse. There's a lot of rich behavior in this task, and it's modulated really based on subject intent. And I'll give you an example of a few of these licks. First of all, we have licks like this that are made to the right, and they go inside the tube because the food is at a position inside the tube that the animal has to make this sort of lick to get. We also can have leftward outer tube licks, for instance, where the food tube is rather full and the animal sort of contorts its tongue in the way that you're seeing here to hit the outside of the tube. These are both are, of course, reward driven. The intention of the animal is to collect reward and retrieve reward. And then we have grooming licks, which are basically everything that does not entail a reward-driven lick. The animal is either cleaning its mouth, cleaning its face. These licks tend to be shorter. They tend to have longer interlick intervals, and they are made less vigorously. Their velocities are typically far shorter, and I'll show you that over here. So this is data from subject M. Red traces. So the first plot is lick displacement in time. I align licks to the onset of a lick, and I show you 300 milliseconds backwards in time, 300 milliseconds forward in time. Just from the oscillatory behavior of this trace, you can see that these licks are indeed rhythmic. Red trace corresponds to rightward licks, blue trace corresponds to leftward licks, and green trace corresponds to grooming licks. And as you can see, the displacement or the total distance traveled by the tongue during reward-driven licks is in fact longer than that of grooming licks. And of course, this is reflected in the velocity as well as the lick angle. The rightwards and leftward reward-driven licks tend to be far steeper in angle, of course, because they're trying to hit these food tubes that are on either side of the animal, whereas grooming licks tend to be sort of shallower in angle. And again, these results are consistent across subjects as well, in that reward-driven licks are longer, more vigorous, steeper in angle than grooming licks. So again, as I briefly mentioned, these animals, when they do initiate licks, tend to make licks really rhythmically and in a long train, right? And here's one example of this taking place. You can see the animal just consistently licking. This is a harvest, or harvesting this reward after making a few trials to accumulate reward in the leftward tube in this case. On the bottom, I have the displacement trace over time, where you can see these sort of peaks of the tip of the tongue, which is what's being plotted there. And really the bout durations, really how long the animal spends harvesting, is highly variable. In one of our animals, subject M, she's far more sensitive to becoming full and sensitive to time, in that her bout durations and total number of licks in her durations will decrease just a little bit. This is not the case for subject R, who is just a machine and will keep busting out trials and keep licking incredibly vigorously, really seemingly no matter how long the task keeps on going, which has been our experience. He seems to be a little bit far more motivated by food consistently throughout the task. Interlick intervals are fairly consistent across these two subjects. They're roughly about 300 milliseconds. These animals lick at about 3 hertz, give or take 0.3 hertz or so. And lick durations are also fairly consistent at about 200 milliseconds on average. So this sort of rhythmic licking in these unconstrained sort of conditioned reward licking tasks is surprisingly fairly consistent across species as well. What I have plotted for you here is subject M's, an autocorrellogram of licks, starting at 0, looking 1 second in the future. The left plot is leftward licks. The middle plot is grooming licks. And the rightward plot is rightward licks. And as you can see, this is very rhythmic. All types of licks are fairly rhythmic as well, but reward-driven licks more so than grooming licks, again, because grooming licks can be very variable in their behavior and trajectories and kinematics. The same can be said for subject R, who is also very rhythmic in its licking. And we see this sort of thing in mice, for instance, in the work of Bryant, that also, when non-constrained and allowed to lick as many times as they want, will lick rhythmically like so, with really sort of a fixed frequency and fixed inner lick intervals. Same can also be said in rats, as is shown by the work of Welsh. Here they plot a autocorrellogram, giving you 300 milliseconds backwards in time as well. And as you can see, these consistent peaks across 3 different rats, fairly similar as well. A lot of rhythmicity to the way that they live. Now, so far I've recorded primarily from lobules 6 and 7 of the cerebellum. I found a lot of lick-related cells, specifically around lobule 6. 14 lick-related cells are in my database right now from lobule 6 of the Marmoset cerebellum. And I've recorded across regions 6AC and 6E, 6E being a little bit more anterior. And I've recorded at about a sort of 1 millimeter lateral range to the right of midline as well as of midline. And I'll show you some data from here. This shows you the region that I'm recording from, lobule 6 over here. And on the right, I show you the model trajectory plans and cerebellum markers that we essentially align our probes to and target. Now, the take-home message that I hope to get across, really just in these preliminary observations that I'll show you, is that Purkinje cell responses vary widely in their response to behavior. And that it can be really difficult to make sense of the function of individual Purkinje cells at the single unit level. It's very challenging. Their behavior is very diverse. Some pause, some burst, some do both. Some are rhythmic during licks in their bursting or pausing, and some are not. And I'll show you some examples of all these things to come. So in some Purkinje cells, the simple spike baseline firing rate is modulated during licking bouts, meaning there's a baseline outside of licking bouts, and then the cell seems to enter this new baseline during licking bouts. And this, in part, can be due to the fact that there's about 50 milliseconds in between licks. And so if say a cell was to burst for a lick, it would remain, its baseline would remain quite high over these rhythmic successive trains of licking. So here's some data from subject M. Okay, this is one cell that I recorded. Here you see plotted on the left is data aligned, simple spike data aligned to lick onset, which is what 0 represents. I plot 1 second back in time and 1 second forward in time. Y-axis is lick number, and y-axis on the left is lick number rather, and y-axis on the right is the simple spike firing rate, essentially the mean of the raster plot that you see. Blue dots indicate simple spike firing. It's just a fairly basic raster plot. Yellow dots are behavioral events, so that's the actual onset of a lick being detected, and as you can see, when it's aligned to 0, it's fairly time locked, and then after that, you do see how successive licks appear. On the right, I do the exact same thing, except aligned to the end of a bout. And what you can see here is a dramatic increase in the baseline firing rate of a cell, looking at after a bout has been initiated and during it, as well as in comparison to before the bout. The bar plots in the middle quantify and will show you essentially this discrepancy in the firing rate before and after. Start corresponds to the average firing rate in the 1 second after a bout's been initiated, subtracted from the baseline firing rate of before, and then the vice versa can be said about the end bout licks. So you can see here that at onset of a licking bout, the baseline firing rate increases and decreases after a bout has ended, and I've seen this in 8 of 14 cells that I've recorded from. This is consistent again across subjects that we have, across both monkeys, and really the extent to which the firing rate is modulated, the baseline firing rate that is, it varies. Some are heavily modulated, some are slightly more modulated, and I've seen some sort of correlation between the baseline firing rate of the cell as is as being a factor. So for instance, if the Purkinje cell's baseline firing rate is at 100 Hertz, during licking, it'll be far greater than if its baseline firing rate was at 50, comparing within licking bouts. And that can be seen here across these two subjects as well. On the contrary, here's another cell from subject R, and we have seen the opposite in that during a licking bout, an animal's baseline firing rate might decrease. And here's just an example of a fairly slight decrease in firing rate. I've seen this in about 3 cells. The top cells were recorded from 6E, which is again a more anterior region of lobule 6. And the cell at the bottom has been recorded from 6AC, which is a more posterior region within lobule 6. Now, if there's a relationship between this behavior that we're seeing and the location that we're recording from, I don't know, and I don't think we can just come to that conclusion just yet. This is just to highlight the diversity in behavior of the cells. Now, within these bouts, and for each lick, simple spike firing can be rhythmic, non-rhythmic, or both, depending on lick direction, which is something that I've seen as well. So here's some data from subject M. This is what I just showed you. It is a cell that, again, its baseline firing rate increases quite dramatically during licking of about 50 to 55 Hertz, and seems to decrease when the animal stops licking. Here is data broken down into lick direction. So what you see on the left is simple spike firing in the same format, except for a time interval of negative 300 milliseconds to 300 milliseconds before and after the lick. The middle plot is that, but for grooming licks. And the rightward plot is that, but for rightward licks. And what you can see here, again, is diversity in the response of the cell, in that For rightward licks, the cell, it bursts quite oscillatory. It's quite oscillatory in its bursting. Grooming licks seems, it also bursts for grooming licks, but far less. And then for leftward licks, the cell seems to stay at a fairly high firing rate, but the bursting is far less rhythmic in comparison to grooming licks or rightward licks. To further capture this, I plot for you a cross correlation from 0 to 1 second of the leftward licks, grooming licks, and rightward licks of the simple spikes. And immediately what you can see is, for the rightward licks, just really beautiful oscillations that seem to indicate that the cell is bursting rhythmically at lick onset. The same can somewhat be said for grooming licks and leftward licks. However, the rhythmicity of that firing is far less impressive and far less apparent, I would say. This is consistent across units that I've recorded from. Here's another cell. Far less of an increase in the baseline firing rate, but still there nonetheless, and then a decrease after licking bouts. You see a similar thing in that, in this case, leftward licks seem to be very rhythmic in their bursting. Grooming licks seem to have no rhythmicity, but their firing rate is increased in comparison to baseline during these licks. And even more interestingly is that for the rightward licks, in this rightward, in the cell that I'm showing you on the right, it seems to be rhythmically pausing for rightward licks. And again, that can be captured in this cross correlation. Here we have 2 rhythmic bursters and 1 rhythmic pauser. And the case on the right is that this discrepancy in whether it's rhythmically pausing or bursting can be seen within the same cell, but for different directions. So perhaps it might suggest some sort of directional based tuning in the simple spikes of this cell. Here's some additional data from subject R. Now, this is a cell that's far less impressive in its baseline firing rate modulation, and I wanted to throw it in there just to express the diversity of the response of these cells. You do see here again, just a very slight increase and a very slight decrease, nothing too crazy when in comparison to the cells I showed you before. However, again, you do see this rhythmicity in the bursting with respect to licking, right? So for leftward licks, the cell again rhythmically bursts, and what's really impressive is that in the duration of the lick, it seems that during the protrusion phase, which happens before 100 milliseconds after lick onset, the cell seems to burst. But then during the retraction phase of the lick, the cell seems to sort of pause and go back to its general baseline during the bout. And again, this can be captured in what you're seeing here. Vice versa is seen in rightward licks, in that at lick onset, the firing rate seems to be sort of at a pause, sort of pausing. But then during the retraction phase of the lick, which happens sort of after 100 milliseconds or so, the cell seems to be bursting. So no consistency there in the response of these cells across phases of a lick and across directions. Here's additional data from subject R. This is one of those cells that seem to pause during licking bouts and then continue their baseline firing rate after licking bouts. Again, small effect, as you can see here. And this cell is really a quite powerful rhythmic pauser. And as you can see here, we have sort of rhythmic bursting on one end and rhythmic pausing on the other end. However, with this cell on the right, it seems to be the opposite of what I described to you on the left, in that the cell, as you can see for leftward licks, the cell will pause during protrusion phase of a lick and then burst during retraction phase of the lick. And the same is actually consistent in its rightward licks as well. So this idea of rhythmically bursting simple spikes of lick-related Purkinje cells is also observed across species. Here I've showed you 3 examples of cells that seem to do this, particularly rightward licks in the top cell, in the middle cell leftward licks, and in the bottom cell, again, leftward licks. And this is consistent to what's seen in Purkinje cells from the mouse cerebellum as well, as was shown by the work of Bryant. Here they plot the lick to spike cross correlation, and you can see it's fairly rhythmic, lots of peaks and valleys that correspond to lick onset and offset as well. These are bursting though. There still seems to be a little bit mystery of these rhythmically pausing cells, and I'd like to maybe dig into these a little deeper in the future as well. These cells that rhythmically seem to reduce their firing rate when an animal licks, not even going into the phase of a lick, but just generally. Finally, what can be said about the complex spikes of these cells? Well, it's been shown that different populations of Purkinje cells will synchronize their complex spike during different phases of lick rhythm. And perhaps if we organize cells with respect to what is happening with the complex spikes. So say complex spike responses of cells, if we were to combine them based on that, maybe this diversity of simple spike may be elucidated a little bit more. Maybe we can make a little bit more sense of that. The work of Welsh and rats does show a nice time lock nature to complex spikes that are seen. And what Welsh does is he combines populations of Purkinje cells that seem to be time locked and synchronous in their complex spike firing. On the left, we have 26 Purkinje cells from rat 1, 29 Purkinje cells from rat 2, and 27 Purkinje cells from rat 3. You can see, again, in the cross correlation, rhythmicity in the firing of the complex spikes. It seems to be fairly time locked to lick onset, actually, perhaps encoding some motor event, perhaps. We see something similar as well in a few of the cells that I've recorded. If you can see here, what I plot to you is a raster plot of complex spikes, which are the red dots. Again, leftward plot is leftward licks, middle plot is grooming licks, rightward plot is rightward licks. And again, the yellow dots rather are the onset of a lick. And I plot for you 300 milliseconds backwards in time, 300 milliseconds forward in time from licks within a licking bout. And what you can see, for instance, in our cross correlations is this same sort of rhythmic bursting, really prominent in the rightward lick cross correlation of this particular cell. Really time locked to lick onset, very much so, and very similar in its look of the cross correlation to that of the work of Welsh. Here's another example of this. And again, you can see this rhythmic bursting that seems to be time locked to lick onset as well in the lick to complex spike cross correlation. So next, I'd like to address just one last observation, which is really, really interesting and really mysterious. And I'm attempting to make sense of it. And it is that some Purkinje cells seem to respond to both licking behavior and saccades. So keep in mind that we are tracking both eye movements, so we know what the eye is doing, and we're tracking tongue movements, so we know what the tongue is doing. And in the analysis that I've showed you based on looking at eye velocities, I've concluded that the cells that I've showed you, during licks, the animal seems to be fixating in space. So not making saccades, not doing the task, none of that while licking as well. So that was licking-driven activity. And that was also made apparent by the increase in the baseline firing rate within and outside of bouts. That's being one piece of evidence that might support that. So here's a cell that one of the cells that I showed you. This is, again, from subject M. We saw that it has fairly rhythmic licking, particularly for rightward licks, and generally a very high increase in baseline firing. Now, let me remind you, this is what the eye trajectories look like. So animals fixate and produce two types of saccades. Now, what will happen if I look at the neural data during these bouts in time where the animal is making saccades and doing the task before and after it initiates licks to retrieve rewards? What I have plotted for you here is data, specifically simple spike firing, with plotted 300 milliseconds backwards in time, 200 milliseconds forward in time. As always, y-axes on the left are the trial number, basically the number of saccades. And rightward y-axis is the simple spike average firing rate, very similar to the rasters I showed you for the tongue. Each one of these corresponds to the direction of the primary saccade that's made prior to the target jump. So for instance, this plot up here is a upward saccade, so a 45-degree saccade. Downwards is 270, and then right is 0, and left is 180, and so on. So this corresponds to the eye trajectory plot that I show you here. And what's amazing is that this cell that seems to also be firing for tongue cells rhythmically for rightward licks is also firing for saccades. And what you can see here, for instance, in this rightward plot is a firing for the primary saccade. And then if we were to look forward in the future, we would see the firing for the corrective saccade that is to come. And this is it's quite mysterious. And what we do also see is direction-based modulation, right? So for generally left, right, and upward saccades, the bursting is it's there, it's fairly apparent, but for saccades made downwards, it's not there. Another cell shows this as well, which is again a cell from subject M. We see here, this is the cell that I showed you that was rhythmically bursting for leftward licks, rhythmically pausing for rightward licks. And again, this is the eye trajectory trace. And what we see is something similar as well. Bursting for saccades, we can see the primary saccades burst because that's what the data is aligned to, as well as the corrective saccade burst in the future, and slightly different direction-based modulation in that these cells sort of from up, down to the right seem to be modulated, but not the ones to the left. And you can really sort of dissociate these two types of activity because the cell, when the animal is making saccades, remains at its general baseline, which is the baseline when there are no licks taking place. And you see these sort of short-lasting bursts. But then the cell seems to be staying at a baseline, whereas for these licks, the cell will enter this new sort of baseline firing rate, and then from there rhythmically burst for tongue movements. So this, again, diversity of behaviors is pretty staggering, and I'm not entirely too sure what to make of this just yet. So these are some of the interesting observations that I made so far, and just to summarize what I've covered in this talk so far, is that, you know, marmosets that we've been working with, they're a very promising non-human primate model to investigate the neural basis of internal models in the cerebellum with respect to fine control of the tongue, and I hope to do more work in this direction. We at the Shadmehr lab, we've developed a lab that facilitates extracellular electrophysiological recordings from various regions of the cerebellum in awake behaving marmosets that are making saccades and licking, behaviors that we're having them do right now. And really the exact functional role of Purkinje cells and licking behavior remains fairly mysterious when looking at it from the single unit level. However, perhaps population level analysis may elucidate a role in, say, temporal regulation of licks, for instance. And that's something that I would like to get into a little bit later on. That is organizing cells based on complex spike properties to see if the simple spikes make a little bit more sense, which is work that has been done in our lab in the saccadic system that I'd like to see done in licking as well. Thank you so much for your time. I'd be happy to take any questions that anybody have.
Well, that was what a fabulous, interesting talk. Lots of questions, I'm sure. Someone want to start?
Yeah, I have a question. Thank you so much, Paul. Really fascinating. On some of the cross-correlation plots, you showed the dynamics of the rhythms, but often it would be like quite a large amplitude, but then it decayed over time. Did that decay reflect any change in the dynamics of the tongue?
Absolutely, yeah. So while the licks are generally nicely time-locked, depending on what the animal is doing with its lick, so for instance, at the beginning of a licking bout, the food might be full, the food tube might be full. So those licks might be a little bit shorter and shorter in duration as well. But as the animal continues to harvest, the licks will have to get a little bit longer to make it into the food tube. So there's a little bit of variation there that when you align these things together might cause sort of a reduction in the peaks. So those reductions in the correlellograms that I showed you, they do reflect what's happening with the tongue and the diversity of the types of licks that are taking place. This is probably something I can control a little bit better in future tasks and such, but that's what explains it, yeah.
Mm-hmm. Yeah.
Thank you for your question.
Is the tongue, I assume the tongue is, has quite a few proprioceptors and muscle spindles. Are there a lot of afferent inputs from the tongue to this part of the cerebellum where you're recording? Was one question. And how about jaw movements? Could this activity be related to jaw movements as opposed to or with tongue movements?
100%. And the elephant in the room definitely is what is happening during licks with the jaw, with the face. These are all things that we have to address. And I think how we can address these is through measurement. We can track what's happening with the jaw. We can, as we do, track what's happening with the face, and I can dive deeper into my analysis just to tease these things apart. So to answer your second question, I don't have a definitive answer, but what we can do is analyze further what's happening with the jaw and the face to try and see how that relates to the responses that we're seeing. And also regarding the anatomy, your anatomical question at the beginning, I don't have a definitive answer. I would have to dig a little bit deeper into understanding the afferents to this region of the cerebellum as well. Yeah. So
certainly for saccades, there is a lot of eye muscle proprioception that goes to the Purkinje cells in that area of the cerebellum. I have to say this, it was a great lunch hour talk. Anybody else want to ask something? Is this in gaze coordinates? Does it, if you move the reward over to the right, left, or up or down, do these Purkinje cells? How is the coding of this movement, these movements, these cells?
Yeah. So what we've seen as one example is that longer licks of larger displacement, have a more exaggerated response from the Purkinje cells. So larger amplitude licks have more, for instance, extreme bursting. And what's interesting is that these animals, they really do learn about where the position of the food tube is at. So when an animal first begins, it may not, as we've seen in some of the animals that we're training now, they can't really make those sort of very dexterous licks to the inside of the food tube. They learn this over time. And what's even more fascinating is that when the animal initiates a licking bout, they're not actually looking at the food tube. They're looking somewhere above in space because those food tubes are fairly low in comparison to the visual field. They're looking at a TV that's positioned upright. The tubes are fairly low, and we've measured these things. And it's tough on the animal to look down and fixate on the food tube. So what we do see is that the animal might stay saccade to the food tube to initiate a lick, but as it continues its licking train, it'll look above the food tube to the order of, say, like 8 degrees or so. 8 to 10 degrees, I think, is the number. So as moving the food tube around, these are things that I would like to play with in the future too. Yeah, that's a really beautiful suggestion. Thank you.
That was a great talk. Thank you.
Thank you.
There is in humans a syndrome where the eyes are moving in rhythmically with the palate. So I wonder if have you recorded in the nucleus ambiguus or the hypoglossal nuclei? I try to because that will really connect with the human. That might be of value.
That would be of value. That would be beautiful to do. Unfortunately, thus far, we've only recorded from Purkinje cells as of now. We would take a little bit of work to record from different regions, but that would be wonderful. Absolutely. Yeah.
Have people recorded from motor neurons in the 12th nerve nucleus? Have people recorded from the motor neurons in the?
Oh, I'm sorry. I've seen a few studies. I've seen a few studies that have recorded from, I believe, cranial nerve 12. I don't know about how extensively though. And this was in the macaques actually. I think it's. I was going to include it as a point of reference in this talk, but I didn't, unfortunately. But yeah, I would have to delve deeper into the literature really just to get at these things.
Seemed to be important. I mean, Dave Robinson started off with the motor neurons, right? Worked back.
Absolutely, yeah. Thank you for that suggestion. I'll definitely implement that.
So, quick question, or I guess clarification for me. Did you mention that I guess the initiation of the tongue saccade is time locked with saccadic initiation in the eyes, or do I have that wrong?
So what I showed you was that the response was time locked. So the licking, bursting, for instance, the rhythmic was time locked to the onset of a lick event. And then what I showed you for the saccades was that those bursts that we saw was time locked to initiation of a saccade, not of a lick. So we.
Oh, I see. Got it. Okay. So have you measured someone licking while making a saccade at the same time?
There are some cases where the animal might do this. Specifically, one of our subjects is more prone to perhaps continuing to make saccades while licking, but that only happens if the food tube is, say, directly in front of its mouth, which is another set. Earlier in our experiments, we had the food tube be very easily accessible. And what we found that the animal was able to do was to make saccades, continue the task, and lick. However, what I've done since is I've pulled the food tubes further. So now it's far harder for the animal to keep making saccades and then also shoot its tongue out and go dexterously into the food tube. Well, the current state of the experiment might make it a little bit hard for the animals to do that. But again, that's another set of things that I can test. That's a really nice point, Dr. Green.
Thank you.
Thank you.
Have you inadvertently or voluntarily made any lesions in this area? And I'm curious if they develop dysarthria of their calls, since they seem to have such very specific speech. Certainly with humans, we associate dysarthria with this very part of the vermis. What about function?
Yeah, we haven't done that yet, or we haven't done any sort of lesion study with these marmosets or inactivation or stimulation just yet. That would, I mean, of course, that would be really fascinating to do. It's just it involves its own difficulties and such. But no, sir, we have not yet, and that would be really nice to elucidate sort of some functional role. It would be wonderful to do, of course. I do know that in mice and in rats, inactivation studies and stimulation studies have shown that the gross movement of the lick is not affected, but there is an effect on the rhythm of the lick and the inner lick intervals of these trains of licks. So licks seem to be a little slower. They seem to be a little less efficient, meaning less volume of reward is retrieved. But the gross licking movement is largely unaffected in the couple studies that I've read up on. This might be, again, further evidence of like temporal regulation of the cerebellum of these like patterned licks.
I just have a question. This was a great talk.
Thank you.
So you showed multiple trials of recording from the tongue, from the Purkinje cells while the tongue was moving. But I wonder if you can, what is your sense about how consistent is the pattern of response in Purkinje cells? Like you record multiple trials, you find the type of response, but what if you just go back another day and record from the same place? How consistent is this pattern of response you get from your single unit recording? I know it's tough, but I mean, do you have a sense already based on your experience?
Generally speaking, yes. So we do consistently record from the same region across weeks, for instance. So we might spend a week going to one particular region and then shift that over to another particular region. But what is consistent in doing this is that the response of those cells are fairly diverse. I know that kind of goes around your question a little bit, but what we do consistently see is that there are cells, even if we were to repeatedly go to the same region, we might be hitting different Purkinje cells. I mean, who's to really be exactly sure if we're targeting that same exact cell? But generally speaking, even cells that are spatially organized close to one another, there's a big diversity of what we're seeing. What is consistent, though, is that we see sort of rhythmic activity. We see this and we can hear it when we're recording very well. And we can, we see that these like saccade-related cells and tongue-related cells and both saccade and tongue-related cells seem to all be packed close to one another. I don't know if there's any significance in where they're located exactly. I hope that somewhat got at your question. I know that was a little all over the place, but it's just really mysterious and we're still trying to get at these types of things.
So to clarify, if a cell is responding rhythmically today, and if you go back tomorrow, would you see the same thing, or the response may change?
Yeah, I don't know if I can answer that because I don't know for certain, even if I'm going to the same region, if that is, and even if the cell that I record the next day is doing the same thing, if that's the same exact cell.
Yeah, that's very tough.
Yeah, it really is. It really is tough to say that. At least doing these sort of extracellular EPHIS recordings.
Paul, this is Sasha. That was really lovely.
Thank you, Dr. Block.
Really nice. Sasha. Yeah, really nicely presented. Of course, just a lot of information, kind of confusing. So, two questions. The first one, did you say what you think drives the complex spikes, and is it different in the cells that also have eye movement responses?
Yes. So what I think might drive these is some sort of encoding of a motor event. That's maybe as specific I can go because I'm not entirely too sure, but what I do see is that in some cells, the complex spike firing is fairly time locked to the onset of a lick. I have seen actually a couple of cells that I didn't include in this presentation that seem to show the same sort of time lock rhythmic nature, but instead of being aligned to the onset of a lick, it's aligned to the max displacement of a lick, meaning that's when the tongue touches its end location. So perhaps that's encoding some sort of sensory event. I just want to collect more cells that show that and better characterize that response.
Of course, right. Okay, the second question is, it's really more of a comment because I can't ask you as a graduate student to do this. This is something that Reza needs to drive you guys to do. Purkinje, the problem with recording from Purkinje cells is that something like 40 of them with many, many different features converge onto a single neuron. And the output of the cerebellum isn't, you're not going to get it from looking, as you know, at an individual Purkinje cell. You're going to get it from looking at the fastigial nucleus neuron. So why not go down there?
That's a near future goal. I think that's definitely a near future goal. We'd like to do that.
Wonderful.
Yeah, I think we have the tools that can facilitate that. We just have to go a little deeper and target it and all that. But that, yeah, really, that would be wonderful to do, Sasha. Thank you for that.
Okay, I think we have another meeting. This was terrific, Paul. Very professionally presented and so exciting. And I'm afraid your PhD is going to take forever to answer all the questions we asked today, but persevere. Terrific talk.
Thank you so much. Thank you, everybody, for your time. I really appreciate it.
All right. Thank you, guys. We'll see you next week.
Thank you. Take care.